Resting membrane potential and action potential. This is a nerve cell, and here is the cell body, otherwise known as the soma. This is the nucleus of the nerve cell, and these branch-like structures are known as the dendrites, which connect to the soma. Dendrites receive electrical impulses, otherwise known as signals, from other neurons, and transfer the impulses into the soma, and from there on into the axon hillock, and then into the axon. The axon hillock starts the action potential, the structure right here, when it receives the signals from the dendrites. The myelin sheaths, which are these purple structures right here, cover the axon to speed up the reaction. They help the body send messages faster to the brain and back to the muscles. These are the nodes of Ranvier. These are the parts of the axon that are not covered up by the myelin sheaths. This is the terminal branches, and at the ends of the terminal branches are the axon terminals. The axon terminals are at the ends of the neuron. This is where the signals are transferred to the dendrites of another neuron or onto a muscle cell. The axon terminals release neurotransmitters, as shown in this picture right here. This is a nerve synapse between the orange, which is a axon terminal, and the red, which is a dendrite. These are the neurotransmitters being released onto the receptors of the dendrite. Resting membrane potential. Resting membrane potential is the charge or voltage across the cell membrane. On the inside of the cell membrane, we have a high concentration of potassium, and on the outside, we have a higher concentration of sodium. Also on the inside, there are large negative ions. This is the cell membrane, and on the cell membrane, we have many potassium leakage channels and a few sodium leakage channels. The way resting membrane potential works is that the potassium on the inside of the cell slowly leaks to the outside because all ions like to go against their concentration gradient. When the potassium ions are on the outside of the cell, this creates a more negative charge on the inside of the cell membrane. It goes down to around negative 90 millivolts. When it gets down to negative 90 millivolts, the sodium channels allow slight leakage of positive sodium ions into the center, and since opposites attract, some of the potassium also leaks back into the center, which averages it out at around negative 70 millivolts again. The second way that a cell maintains its resting membrane potential state is through sodium-potassium pumps. Since sodium has been leaking into the cell and potassium has been leaking out of the cell through the leakage channels, the cell needs a way to return back to its resting membrane potential. It does this with sodium-potassium pumps. These sodium-potassium pumps are working against the concentration gradient, so they require the energy from ATP, which binds at the ends of the sodium-potassium pumps, to pump 3 sodium ions out of the cell for every 2 potassium ions it pumps back into the cell. This returns the cell to its resting membrane potential state. Action potential. Action potential is the change in electrical potential associated with the passage of an impulse along the membrane of a nerve cell. There are 4 stages in sending an impulse. Stage 1 is resting membrane potential. Stage 2, 3, and 4 are action potential. This is an impulse chart. Stage 1 is resting stage, depolarization, repolarization, and hyperpolarization. When a nerve cell receives an impulse through its dendrites from another nerve cell, if the impulse is great enough to make the voltage or the charge of the nerve cell to rise to negative 55 millivolts or above, then the nerve cell will fire. Negative 55 millivolts is what we call threshold. All the cell needs to do is reach that level and it will fire. It will create an impulse. So once the impulse is created, we have the nerve cell depolarizing. This is called depolarization. And in the depolarization stage, the voltage-gated sodium channels open up and sodium rushes into the inside of the cell. This vast amount of sodium positive sodium ions rushing inside the cell creates a very positive atmosphere inside the cell. And so, as you can see on the chart, it rises up to around positive 30 millivolts. Stage 2, repolarization. In stage 2, we have the voltage-gated sodium channels. And the voltage-gated sodium channels are opened by depolarization. When the cell depolarizes, the voltage-gated channels are opened and the potassium rushes through the potassium channels out of the cell. And with a great amount of positive potassium ions leaving the cell, the inside of the cell becomes drastically more negative. During hyperpolarization, the sodium channels are closed by an inactivation gate, while the potassium channels stay open. This causes the cell to go down to around negative 90 millivolts before it goes back up to its resting membrane potential stage of negative 70 millivolts.